Hello, and welcome to the latest of the Kavli Foundation Spotlight Live webcasts, which offer a chance to hear from scientists on the cutting edge of discovery. Today, we are going small. We'll be talking about microbiomes, communities of one-celled organisms and viruses that form the world's smallest ecosystems. Microbiomes exist within our bodies and virtually everywhere else on the planet. Without them, life on Earth would be impossible. And it turns out that microbiomes have a great deal to teach us. Their biology positively brims with novelty, green chemistries, and life-saving pharmaceuticals. They might be able to show us how to grow crops without fertilizers or pesticides, reduce infection in hospitals, and fight such autoimmune diseases as asthma. Yet despite their small size, these microbial ecosystems can be extremely complex. A few grams of soil or ocean sediment may hold thousands or even hundreds of thousands of different species of one-celled organisms and viruses. So the question is, how can we learn more about these little-known ecosystems? I'm Alan Brown, and I cover nanoscience for the Kavli Foundation. I'll be moderating today's discussion. With us to answer your questions are two leaders in applying nanoscience to the microbiome. Eoin Brodie pioneered a device capable of surveying the bacterial species present in a microbiome. He is currently developing ways to combine data from many different types of measurement tools into a more coherent picture of these ecosystems. He is a staff scientist at Lawrence Berkeley National Laboratory. Jack Gilbert manages the Earth Microbiome Project, which is characterizing the microbial diversity of our planet. He is also studying hospital microbiomes and is working on ways to embed bacteria in nanostructures to fight autoimmune disease. He is a researcher at Argonne National Laboratory and the University of Chicago. Now, before we begin, let me remind you, if you have a question you'd like to ask, please send it to Kavli Foundation by email at info@kavlifoundation.org or by Twitter using the hashtag right under my name, hashtag KavliLive. So, let's start with an obvious question. Jack, Eoin, I gave you a 5 or 6 word definition of a microbiome. Is it a good one?
Yeah, I think that it works pretty well. It's a collection of organisms within an ecosystem, and you can think of the ecosystem of microbes just being like an ecosystem that you would see in terrestrial systems on Earth, like a tropical forest or a grassland or something like that. Collection of organisms working together to maintain the function of a system.
Yeah, so this is the microbiome is the bacteria, the archaea, which is like another single-celled type of bacteria, and the viruses and the fungi, anything that's a single-celled organism, and they come together as a community of organisms, just like a population of humans in a city, to do different roles that have then an emergent property, something which the community does to facilitate a reaction or response in an environment.
So how complex can these microbiomes become? Are they like tropical forests? Are they more complex, less complex?
The diversity of microbial life, the diversity of microbial life it pales a eukaryotic life, that's all of the living animals and plants that you can see, into insignificance. I mean, these bacteria, these archaea, these viruses, they've been on the Earth for 3.8 billion years. I mean, they are so pervasive. They've characterized they've colonized every single niche on the planet. They shaped this planet. I mean, the reason we have oxygen in the atmosphere is because of microbes. Before they started photosynthesizing light into biomass, the atmosphere was mostly carbon dioxide. The reason the plants and animals on this earth exist is because of bacteria. And the diversity of all the plants and animals, everything you can see that's alive today, that you can see with your eyes, that's a drop in the proverbial ocean of diversity that the bacterial, the microbial world actually contains.
Yeah, we tend to think of the Earth as being a human planet, and that we're the primary organism or the alpha species. But we're really passengers. We're blow-ins on a microbial planet. We're recent additions.
Excellent. And you both wax so poetic about it.
Makes me
So,
excited.
so then here's the million-dollar question. Why is it so hard to understand what goes on in these ecosystems?
I think, well, Jack alluded to it there. The first problem is they're very small. We can't see them. It's very difficult to understand how things work when you can't see them. So tools are needed to develop to be able to see these organisms. We also can't grow them. It's very hard to bring them from the natural ecosystem into the lab. Probably less than 1%, depending on the ecosystem, maybe a little bit more in the human microbiome, can actually be cultivated on growth media in the lab, grow them on petri dishes so we can do experiments and understand what functions they actually carry out. So that leaves 99%, or the vast majority of microbes on Earth in most ecosystems, unknown to us, apart from their DNA signatures and things like that. Jack has pioneered these types of analyses, and when you look at the DNA signatures from these environments, there's all these new organisms, these new proteins, these new functions that we really have never appreciated before. And this has been called Earth's microbial dark matter. So just like dark matter and energy in the universe, this has been unseen to us and unknown to us, but it's extremely important for the planet to actually function and for us as humans to function.
Jack, before you jump in, I just want to follow up, Eoin. What makes it so hard to grow these microbes in a petri dish? Either of you can answer, actually.
Yeah, well, I'll take a stab at it first, but they're very fussy. You can think of it that way. They don't like to eat the food that we give them in many cases. They eat things that we don't know that they can eat. They breathe things that we don't know that they can breathe. We breathe oxygen. They breathe oxygen, but they'll also breathe nitrate, iron, sulfur, even carbon dioxide. So, and getting the concentrations and the combinations of what they eat and what they breathe is very difficult. In some cases, not only even if we can work that out, there may be something that they need to get from another microbe, another member of the biome, another member of the ecosystem that has to supply an essential nutrient or cofactor for them to grow. So getting all of those possible permutations and combinations right is extremely challenging. A lot of people are working on it, and there's a lot of expertise being put at this, but it's extremely difficult and complicated.
That's an interesting point. Sorry, Alan. I liken it to having a baker. If you have a baker in a human community, the baker needs somebody who makes the flour. He needs somebody who can provide him with a bit of yeast. She needs someone who can buy the bread. They exist as a network of individuals living in a community. If you take the baker out of the community, they can't make the bread, and they can't sell the bread because they can't make the bread, and they become not a baker. Taking a microbe out of the context of its community reduces the likelihood that it will be able to do the roles and tasks which it does in that environment. So it's almost like you don't want to try and culture these things. You don't want to try and grow these things in isolation. Because when you do grow them in isolation, while it makes our job as microbiologists easier, it's also much more difficult to understand what they're actually doing in the world that they live in the environments, in your guts, in the oceans. We can't figure out what they're doing when we look at them in isolation because they are basic community players.
So this would be like trying to understand what goes on in New York City by only looking at the crowds.
Yeah, or by only looking at cab drivers. Could you understand New York City by only looking at cab drivers? Actually, maybe you could. I don't know.
You have a very different perspective of New York City.
Well, I've asked you this question before, and both of you laughed at me. But let me ask it again. What are some of the tools that we can use today to look at microbiomes? Is there a state of the art?
So I'll take on that. I mean, this is a very dynamic, evolving field. It is not a field which is ever seems to rest on its laurels, to use a vernacular. To understand microbes, we have a couple of tools which are available to us. One of those tools is genomics. So we can sequence the DNA that comprises their genome, just the same as we've done for the human genome. We can do that for bacteria, we can do that for archaea and viruses and fungi, right? The second one is we can look at the RNA. That's a transient molecule that translates what's in the genome and makes into the protein which creates the cell. Okay, and that's useful. It tells us which genes are being turned on and off when we put those microbes under different conditions. Then we have the proteome. These are the proteins that actually make up the cell. Those are the enzymes that enable the organism to interact with its environment, to consume its food, to make it, carbon dioxide, to respire the oxygen and iron, etc. And then you have the metabolome. And the metabolome is the metabolites, the chemicals, the molecules that are generated and consumed by that living organism. That's the food and the waste products of the bacterial cell or archaeal cell or fungi. So those, the genome, the transcriptome, the proteome, and the metabolome are four of the tools in our toolbox that we have to actually examine the microbial world. But they are by no means the limit of our tool or our goals. We have ambitions far beyond just examining those components. Eoin has some is developing some of these, and maybe Eoin, you want to jump in now?
Yeah, I'd add to that. I mean, the challenge of understanding a microbiome and even individual microbes is the scale, that they're just so small. They're complicated and small. So understanding where they are and the activity of them, so either the transcripts or the proteins or the metabolites at the scale that they exist, is extremely challenging. So all of these technologies that Jack mentioned have been developed, with bigger cells in mind or bigger or larger organisms in mind. So scaling those down to deal with the size of microbes, but then increasing the throughput of them to deal with the complexity of microbes is a huge, huge challenge. When we look at the activity of an ecosystem, let's say you look at we're talking about a tropical forest, and you look at the distribution of trees, and then you look at the distribution of animals, and you look at the association between vegetation and animals, for example, and insects. You have space in mind. You think this lives near this, it interacts in this area. So there's an interaction, there's a fundamental association between those organisms, those members of an ecosystem. The way we typically look at microbiomes, this is changing, but the way we've typically looked at them is we take what would be the entire forest, and we mash up that entire forest in a blender, and then we take all of the DNA and we sequence the DNA, and then we look at the proteins, we look at the RNA and metabolites, and then we try and go back and say, this tree is interacting with this insect, whereas in reality, that tree is hundreds or thousands of kilometers away from that insect and they never see each other. So we have the same problem in the microbiome, in that many of the organisms, when we mash up and we get rid of that spatial structure and association, we lose that importance of space in terms of facilitating interaction. So really, I think that the next wave in microbiome research has to target this microbial activity and interactions at the scale of the microbe. Do they see each other? Do they interact? And how do they interact? What are the chemicals that they exchange? Under what conditions? I think that's the real challenge, and that's why we're talking to the Kavli Foundation. That's where Nano comes in.
I was going to say, this is a lovely transition to my next question. Where does Nano come in? And I can give you an example. For example, Eoin, could we use some of the same nanoscale probes that are being developed to study the brain to, say, investigate microbiomes in water or soil?
Yeah, I think there's some interesting parallels. I mean, you think of the brain as this extremely complicated network of neurons, and the Brain Initiative is attempting to map those neurons and to follow activity at those neurons and synapses, etc. The microbiome is a network of interacting organisms that turn on and turn off, and the connections and the structure of that network is extremely important to the functioning of that system, just like it is for the functioning of the brain. So for the Brain Initiative, people got together and they said, well, what do we need to be able to do this non-invasively in real time to look at electrical charge and electrical flow through neurons. And they came up with some technologies, these potentially, you could call it remote sensing, but remote sensing on a very small scale. And that's looking at how the system changes. So to do this non-invasively, one approach is to use imaging, external imaging, and another approach is to embed sensors. In the Brain Initiative, some sensors are being developed here at Berkeley Lab and elsewhere that use RFID, and they're radio frequency powered. And we talked about this before. These are the types of things that are used to track shipping containers, the types of things that are used to track goods in department stores and things like that. They both transmit information and harvest energy from radio frequencies. So they're autonomous devices. And now I think that the challenge is coupling that technology to sensors. So sensors that are sensing something in the environment, taking that information and sending that information autonomously, no batteries required, to receivers. And then if these sensors are distributed in an intelligent way, then just like GPS, you can triangulate where that information is coming from. So how could you use this to understand a microbiome? Well, this is still relatively large scale. These sensors are being developed. They're about a millimeter in size. Pretty small for us, but very large for a microbe. So you could think about this in soil. Let's say we want to understand what happens when a root grows through soil. It stimulates microbes. There are 10 times more microbes near a root than there are away from a root in soil, and different chemistries and different functions happen. It's very important for the nutrition and health of a plant. If you could distribute these sensors in the soil and have them sense things like carbon from roots or oxygen that gets consumed by microbes, things like that, then you could build a three-dimensional picture of how the soil microbiome is changed and altered as a root goes through the soil. That's one example of how new advances in other fields driven by nanotechnology could be applied to microbiome.
That's spectacular. And one of the things that interests me is, of course, these would be based on semiconductor chips. Mm-hmm. So you could take a wafer, make a lot of them, right? And then distribute them cheaply right into the soil.
Yeah.
And get a picture you couldn't get any other way.
Yeah. There's a field now, we have predictive medicine. There's also a field growing called predictive agriculture. It's like personalized agriculture, where fertilizer addition, for example, in a field would not be uniform. You would deliver the fertilizer where it's needed. You would irrigate the field exactly where it's needed. So you have this massive network of distributed autonomous sensors, and that would allow us to more efficiently use fertilizer and use fertilizer in ways so that it wouldn't be leached or lost from the system and cause water pollution and things like that. These are just some examples, not the microbial scale, but these are microbial processes that control the availability of these fertilizers and control their flux from the system.
Good. Hold that thought. We'll come back to it in a few moments. In the meantime, Jack, you did some work on studying the evolution of microbiomes in a new hospital to see how they evolve and affect the spread of disease within the hospital. Could you tell us what you did or are doing, and how nanotechnology might help future efforts?
Yeah, so the microbes that exist in a hospital have been a focus of clinicians and medical researchers for a couple of hundred years, since we uncovered the fact that bacteria might actually be causing disease. We've been trying to kill those bacteria in hospitals, so eradicating microbial life as much as possible. And that's changing. That paradigm is shifting to one where we're more interested in trying to understand how bacterial communities in a hospital may be facilitating disease spread, maybe facilitating antibiotic resistance, or maybe facilitating health. So we've been going into hospitals and in a very, very high temporal resolution, exploring how the bacterial communities in those hospitals change over time. Right? So looking at the scale of hours to days, we're trying to understand how when a patient moves into a new room to have an operation or to undergo a procedure, how the microbes that are in that room already affect the outcome of their stay in the hospital. how it makes them either healthier or sicker. What's interesting about this is we've been obviously cataloging the microbes at these very fine scales, and we see that there is exchange between the bacteria in the patient's room and the bacteria in the patient's body. But we've also uncovered the fact that the vast majority of bacteria that we would normally associate with so-called healthcare-associated infections, or these pathogens that people are thought to have acquired during their hospital stay, the vast majority of those instances appear to be bacteria that the patient brought into the hospital themselves. They're bacteria which we have inside us. And remember, we have 100 trillion bacteria living inside us. They weigh about 2 pounds, about the same as the brain. So if you think that the brain initiative is important, well, maybe the microbiome initiative would also be important because it weighs about the same. And that microbiome inside us has a lot of players, a lot of them are friendly to us. Most of them are friendly to us, but they can turn on us. I liken this to a riot spreading in a city. If you have a lot of disaffection, if you have a lot of poverty, if you take things away from people, they will generally rise up and try and overthrow the very thing which was supporting them in the first place. And microbes are the same way. We take a patient and we put them in a hospital, and we give them antibiotics to kill the bacteria. We give them radiation therapy in order to kill the bacteria, and then we cut open their intestine and expose the bacteria to oxygen, which they don't like in this case. And we stitch the gut back up and look at the bacteria inside it. We see that bacteria that were previously friendly have started to riot. They've been insulted so many times by the treatment that patient got in the hospital that they've decided they've had enough, and then they go and attack the host to regain the resources which have been taken away from them. And this is very important. Understanding the microbial treatment, understanding the treatment of patients, the hospital stay from the microbes' perspective, is helping us to design better ways to treat patients, design better ways to reduce the likelihood those pathogens, those microbes inside us, will rebel against our bodies and attack us and make us sick. And so nanotechnology is helping us to get a finer and finer scale of visual resolution to understand exactly at what time point during a surgical procedure a bacteria becomes rogue and starts to attack the host, and what molecular mechanisms underpin that. So, we found, for example, this is one final example, it's a great one. We found by looking at particular biosensors, so molecular nanotech biosensors that we can place in the gut, that when the phosphate level, the phosphate is a very important molecule. It's used to create all the DNA and proteins in our body and in the cells of those bacteria. When the phosphate level drops below a certain threshold, the microbes turn on mechanisms to acquire phosphate. They turn on mechanisms to grab phosphate from their environment. And where's the best source of phosphate? It's in the gut lining of the host of the human. And so they migrate to the gut, and then they start to break down the human cells. And we experience that as a severe pathogenic infection, and which often kills us. And so understanding that, we're developing mechanisms to release phosphate at exactly the right time during surgery to prevent those bacteria from ever experiencing that phosphate reduction. And so then you need little micro phosphate releasers. And so we're developing nanotech scaffolds which contain phosphate and placing those into the gut during surgery to reduce the likelihood that the pathogens will become pathogenic.
Not only is that interesting, but it feeds right into a question that one of the viewers just sent us about whether we can adjust microbiomes so that they can target diseases and other human conditions. Can they go beyond just adjusting pH and do something more aggressive?
So, I mean, the win story in this one, the one where we've had the best success, is in treating chronic infections such as Clostridium difficile, also called C. diff. So C. diff infections are chronic gastrointestinal infections. In those instances, we've used a shotgun approach where we've taken the bacteria from a healthy person and we've transplanted them into somebody with a chronic C. diff infection, and that's overridden that C. diff infection and allowed that person to get a healthy microbiome established in their gut so that they're no longer sick. The Chinese did this back before about 2,000, 3,000 years ago. They called it yellow soup. They fed the stool from a healthy person to a sick person and made that sick person healthy. We've just rediscovered this process and are now applying it in a more clinical practice. But that's a very untargeted approach. What we're trying to do with programs such as American Gut, which is our research arm, works with the public, and also programs associated with autism and Alzheimer's and Parkinson's patients, is to identify the specific bacterial community members which are either absent or overgrown in those patients suffering from those conditions, and explore how to adjust their abundance. So maybe we add one back in, or maybe we need to knock one back in order to make that person healthier.
Good.
Yeah.
We'll come back to that in a moment. In the meantime,
Could I add
want
something to that?
Oh, sure. And then I've got a question for you also.
Okay, there's an interesting analogy, I think, in that this is what we're doing in fecal transplants, in this case for C. diff, is the same as, it's called restoration ecology. And people do this in natural ecosystems. You go and you take out an invasive plant species, you weed it out of a system, and then you go in and you plant in other plant species to outcompete that invasive plant species. It's the exact same process. So the same ecological principles and ecological theory that's used in restoration ecology can be used in medicine. And in some cases it may not be as simple as trying to remove one organism or adding in one or two organisms to remove that other organism. It may be a community function. It may actually need that complexity to be able to outcompete the organism that's causing the disease.
That's a really interesting point. Both Eoin and I are microbial ecologists at our core, but I started out in marine microbial ecology, and now I work in soils and in plants and in humans and in disease. And Eoin does the same. You know, we can apply the ecological principles of microbes to any environment because microbes are everywhere.
Good. So, Eoin, we have two questions for you from the audience. One of them involves agriculture. Could nanoscience help us alter microbiomes in ways that alter how we grow, fertilize, and protect plants from pests?
Yeah, that's a great question, and I think a really timely one as well. We're at 7 billion world population, heading to 9 and towards 11. We're gonna run out of fertilizer. We're gonna run out of space to grow food. We're running out of water. We're in a severe drought in California. These are all challenges globally in feeding a global population and providing fuel for a global population. Things that we can do, things that microbes can do in nanotechnology, are mainly revolve around improving the resistance of plants to stresses. Those stresses could be drought stresses. Microbes can help plants acquire water. Fungi, mycorrhizae can increase the root system, improve drought tolerance, and improve nutrition. And we can also identify bacteria that can produce fertilizer in the plant or near the plant. So bacteria that can take nitrogen from the atmosphere and fix nitrogen potentially can offset the use of nitrogen fertilizer that takes a lot of energy and cause a lot of pollution. Bacteria can also mine critical minerals from the soil. We can have bacteria growing with the plants that dig up phosphorus, just like Jack was saying. Bacteria have these strategies to acquire phosphorus from the environment. We can choose those bacteria so that they mine more phosphorus than they need and supply that to the plant. All of these things would reduce our reliance on mining phosphorus from strip mines, for example, or using 5% of our world's energy to produce nitrogen fertilizer. I think that's a big challenge. So nanotechnology, I think, as I mentioned earlier, can be used to characterize the organisms, understand how they work, but also build in sensors into systems to identify when nutrients are limiting, so you don't need to use nutrients and fertilizer in a very inefficient way. You use it in a very targeted, specific, and a much more sustainable way.
To take a step beyond that, is it possible, for example, to control pests this way?
Yeah, and actually that's been done for a long time. Now there are GMO crops out there that have been developed, and they have taken genes from microbes that are used to kill insects, for example. That's been used. This could be carried out in a more natural way as well, while having these bacteria growing with the plants and potentially inhibiting insects from grazing and feeding on plants, for example. We can learn a lot from nature. Nature has already developed these strategies for pest control, and we can learn from that and design it in a more maybe controllable and intelligent way. Not more intelligent than nature, of course.
Okay, one other question from a viewer for both of you. Is it possible to make an artificial microbiome community do a particular task?
Yeah, we've actually been working in that area, trying to create what we call a simple minimal community. This is a community of organisms which performs a task that we're interested in for whatever reason. One of those has been trying to create acetate or generate hydrogen or create butanol as a potential biofuel source. And so looking at microbes which grow on the surface of cathodes and take raw electrons from those cathodes and integrate them with a carbon dioxide source such as flue gas from a factory, we can create a community which drives its metabolism towards a set goal. And that takes mathematical modeling approaches, so metabolic modeling, trying to actually, in a computer, synthesize how these microbes interact to release a certain product. So in that sense, you need nanotechnology to be able to sense the metabolic relationships which exist between those organisms, so you can engineer that community towards producing a particular product. That's very important for our biotechnological aspect.
Good,
Yeah.
Owen.
Yeah, I got to turn that question on its head. I'd like to be able to take a natural microbial community and stop it doing something in certain cases. Let's say, for example, you've got cattle, livestock, and they're a significant source of global methane that contributes to global warming. Part of that is because of their diet and because of what they're eating, and that there's excess energy there, and that excess energy is resulting in a lot of hydrogen being produced, which results in a lot of methane. Cows release a lot of methane. So could we go in and use synthetic biology approaches or chemical interference approaches that are specific and targeted to stop the production of methane, to alter the balance of the cow's rumen, the cow's gut ecosystem, the microbes that are in there, and not only inhibit the methane production, but improve nutrition to the animal, because it's microbes that control the flow of energy to the animal from the food that they're eating. So going in to a complicated system, but specifically tweaking that system for the benefit of the animal, but also for the benefit of the planet. I think that's an interesting challenge, and there are people working on that.
No, I'd like to take that exact system and apply it to coal in order to make more methane that we can then capture and pump into people's homes as a biofuel.
Good. Now, we're getting close to the end, but I did have a question from a viewer, particularly. I think Jack, you're the guy to answer this. She had heard of experimental treatments that involve implanting healthy gut bacteria into people with autism. Why might this work, and will this be something we see soon?
So very simply, the bacteria in our gut have an impact upon our neurological behavior, the way we behave, through our immune system. So they elicit a certain immune response in our gut, which feeds back on our nervous system to create a certain characteristic behavior in our brain. We've known this in animal models for a number of years now, and we're just starting to understand how neurological diseases such as autism, such as Parkinson's, and conditions such as Alzheimer's are attributable to a disruption in the bacterial community in somebody's intestine. And so, yes, there's been several experiments. A very low number of children have been experimented upon, but several in South America and a number in Australia, where the children have had a fecal microbiome transplant. So they've had a healthy microbial community transplanted into that patient. The results are variable and not exactly something you'd want to try at home. but they do hint in some instances to a favorable outcome, i.e. the child's neurological disorder is lessened or significantly reduced. But groups out at Caltech are generating probiotics, so particular bacterial species which they hope to be able to add to a child's diet or put into a capsule that can be swallowed, that could be that seem to have a benefit in reducing the neurological abnormalities associated with autism to significantly alleviate those conditions. Now, again, that's very early days.
Now, let me ask you one other question, because I know you're also doing some work that is related to this, but very different from implants. Your goal is to encapsulate microbiomes in some sort of nanostructure, and then apply this to homes or offices in ways that you might be able to enable people to develop the immune system they need to resist these problems.
Yeah, we're working in animal models at the moment, but imagine we're creating structures that these animals can interact with. But imagine I built you a building that was biologically alive, where the walls were deliberately teeming with a healthy microbial community. And I have no idea yet what healthy means. I have some idea, but it's very limited. But essentially what we're doing is creating structures, 3D printable structures, that are impregnated with certain nutrients. We're working with Ramil Shah up at Northwestern University on this, to create a 3D structure which allows that community, that bacterial community, to thrive. And what happens when we see that is that we can introduce these structures into a mouse's cage, and the bacteria that are associated with this plastic surface colonize that mouse and reduce certain abnormalities that we see in that mouse, such as an allergy response. So we've been growing bacteria which can produce a chemical which they release into the gut of the mouse once they've colonized it, that reduce the likelihood of that mouse having a food allergy. And working with Kathy Nagler at University of Chicago, we're hoping to prove that we don't have to pump kids full of probiotics. We could just redesign homes and offices and maybe even creches so that the children will get an appropriate microbial exposure now when they're growing up, in a way that would mirror how they would have grown up if they were in a natural ecosystem. Hopefully that will be the future of architecture.
And as a possible alternative, we could send our kids outside to play a bit more.
Oh, not that. Very good. I must say, Eoin, Jack, thank you for joining us today. This has been a true magical, and I mean magical, mystery tour of one of Earth's great mysteries, right? The black matter of Earth, natural microbiomes.
Dark matter. Mm-hmm.
Yes, I'd like to thank all of viewers who joined us today, and especially those who sent us questions. And if you want to watch this webcast again or share it with your friends or colleagues, it will be available almost immediately on the Kavli Foundation website that you're viewing now, and also on our Kavli station on YouTube. And if you'd like to learn more about future webcasts or just keep up with nanoscience news, please follow the Kavli Foundation on Twitter. Our handle is @Kavli Foundation. And once again, thank you everybody for watching. Eoin and Jack, thank you for joining us today.
Thank you.
Bye now.